Hi, I'm Dr. Edwin George, and I'm going to speak to you about alpha-synuclein and beyond, drug targets for MSA. In some respects, this is just a summary of some of the current clinical research going on for treatments for multiple system atrophy. And I have to include this disclaimer. I do work for the FDA, but this talk reflects my views. It does not represent the views of the FDA. And also all of the information I'll be presenting comes from public sources, nothing from the FDA's available information. I have nothing to disclose in terms of any conflicts of interest. And so we'll start pretty much where I started my talk last year. It looks simple. We know that Lewy body and other synuclein pathologic changes spread through the brain from brain cell to brain cell. And we know that a misfolded protein, alpha-synuclein, is the agent, and it can fold and aggregate in different ways to cause different diseases, such as Parkinson's or MSA or dementia with Lewy bodies. And the misfolded and aggregated alpha-synuclein damages brain cells. It does look simple, but our job is to make things as simple as possible, but no simpler. This is actually a misquote, an oversimplification of something Einstein actually did say. What he said is more like, it can scarcely be denied that the supreme goal of all theory is to make the irreducible basic elements as simple and as few as possible without having to surrender the adequate representation of a single datum of experience. A bit harder to swallow. But the key thing here, of course, is that no simpler relies on this include every datum of experience. So as we accumulate more data, we do have to change our simple explanation to accommodate new information. And of course, this is the long-standing now point of view of the Lewy body pathology, particularly as in Parkinson's disease. Brack and his co-workers found in a series of autopsy cases that there seemed to be a spread over time of the Lewy body pathology, starting in the brainstem, the dark red at the top image of the brain, and gradually spreading out to the edges of the brain and around to the outside of the brain. The same type of spread, though a slightly different pattern, has been demonstrated. So this slide shows the progression of glial cytoplasmic inclusion pathology, which is what we see in MSA. And we have a top row of images progressing from left to right of MSA-P with Parkinsonian symptoms predominating, and MSA-C with cerebellar symptoms predominating. And you can see that the colors get more intense and things spread around as the disease progresses. And the big difference is you have two areas that are black with the MSA-C, whereas the MSA-P, by the end, the black is still centered in the brainstem. The brainstem is where the Parkinsonian symptoms mostly arise from, and the other area that's black is the cerebellum hanging off the back of the brainstem. And of course, the key to this in our simple explanation is the prion mechanism of protein misfolding. And as you can see at the top of the slide, proteins are made as little chains of amino acids, and these can fold up in different ways. And there's slightly different colors along the length of the amino acid chain. And some parts of it stick nicely to other parts, which influences how it folds. It does not actually fold into these nice geometric shapes that we see here, but rather it folds into different shapes that the artist has chosen to make geometric shapes to make it easier to portray. But some of those shapes don't tend to clump together at all, but other shapes can aggregate, as we see with the green and the yellow, on either side of the image. And they can aggregate into small clumps, which you see about halfway down the image. But the ones that we're concerned about can go on to further aggregate into fibrils along thin aggregates of protein, as you see at the bottom of this diagram. So this is the idea that we can have, and that again, the different folding, the green ones versus the yellow ones, might, for instance, reflect MSA versus Parkinson's disease. And this mechanism seems to have held up fairly well. And you can demonstrate, as you see in the top article represented here, that you take the alpha-synuclein fibrils from misfolded alpha-synuclein and inject them into an animal, and you can induce more proteins to misfold in that animal, resulting in Lewy body pathology. And that leads to synaptic dysfunction, disruption of the connections between nerve cells, and it leads to neuron death. And at the bottom, we have the cell-to-cell transmission of pathogenic proteins in neurodegenerative diseases. These misfolded proteins can, in fact, spread from cell to cell in the brain, similar to the patterns we were looking at a moment ago of the pathology spreading through the brain. So the spread of the misfolded proteins, acting as a template for further protein misfolding. I didn't really emphasize that when we were looking at the little geometric shapes, but the idea is that once it starts misfolding, it can actually induce further protein chains as they're made to fold up in the way that fits for binding together and aggregation. And so this becomes a potentially multiplicative chain reaction. Once you have cells with misfolded protein in them, it not only spreads to other cells, but it keeps encouraging more misfolding and more aggregation of abnormal protein, which, as we just said, can cause disruption of the connections between nerve cells and actually cause the nerve cells to die. And one problem with this is that it may well turn out that we're able to slow it down by using drugs to interfere with the aggregation, or to help break down the aggregates or otherwise take them out of play. But it's likely that it will prove that we cannot totally reverse or eliminate the misfolding. There will still be some degree of ongoing protein misfolding and aggregation and damaging nerve cells if we focus on just trying to prevent that process. So this is sort of like trying to get a genie back in a bottle. Once it's started, it's not clear that we'll be able to totally prevent it. It's possible, we can hope, but it seems most likely with the drugs that we're studying these days that we're going to slow down this process of protein aggregation and damage to nerve cells. So here is a diagram that shows some of this pattern. And actually this, the article that I took this diagram from is mostly about these gray boxes, which list some of the drugs that are being studied to interfere with the progression of particularly multiple system atrophy. And as you can see, sort of centered on the screen is this orangey tan cell that's labeled oligodendrocyte. And within the oligodendrocyte, which is where the abnormal protein production and misfolding and so forth takes place, you see a cycle around its red nucleus of mRNA down at the bottom, and then going around clockwise from the bottom, you get alpha-synuclein monomers, and then you get alpha-synuclein oligomers. That's a few aggregated alpha-synuclein molecules, and eventually fibrils. And then continuing around clockwise, it shows that the cell may be able to break down some of those aggregates as they form, rather than letting them get free and cause damage to other cells. And so some of these gray boxes arranged around there are showing different agents that interfere with different steps of the process of forming misfolded alpha-synuclein and getting it aggregated and eventually causing fibrils. And so there's another gray box sitting up a little higher to the left side of the screen that says immune therapy. And that's showing us these aggregates, the oligomers, outside the cell being bound by these little green Y-shaped things, which represent antibodies. And so one approach is to have antibodies bind to the misfolded aggregates of alpha-synuclein once they're outside the cell. And this could take them out and decrease the ability of the aggregation process to spread from cell to cell. It's a type of therapy that has proven to work fairly well. It can work to some degree in Alzheimer's disease. And so there's a lot of interest in using the same sort of therapy in the alpha-synucleinopathy diseases, including multiple system atrophy. So this table shows some of the completed and ongoing clinical trials in multiple system atrophy that specifically target alpha-synuclein. At the top, we have ION 464, which actually is an antisense oligonucleotide that binds to RNA and discourages it actually being used to produce more protein chains. So that would definitely slow down the process. And that's a random controlled trial that's ongoing. It's in recruiting at this point. And then below it, TAC 341 and LU-AF 82422 are passive immunization. So this is giving people intravenous antibodies that could bind the alpha-synuclein. And those are also randomized controlled trials that are no longer recruiting, but are actively gathering data on the enrolled patients. And then we have a somewhat clever approach of immunizing the patients. So they make their own antibodies. PDO1/03 is actually an agent that immunizes patients so they make antibodies against toxic alpha-synuclein forms. And this is a trial that the very initial trial has been completed, and it's safe and well tolerated. And it did induce an immune response. Patients made antibodies against the alpha-synuclein target. But this is an early phase 1 trial, so we don't yet know whether this drug can cause a real change in the progression of multiple system atrophy yet. Similarly, UB312 is another agent to actively immunize patients so they make their own antibodies against alpha-synuclein fibrils. And this is an active trial that's gone beyond the recruiting phase, and we don't have results yet. So some of the other things that have been tried, rifampicin comes next and could actually potentially inhibit the formation of the fibrils. But this study was prematurely terminated because it seemed to be ineffective. Similarly, and I won't try and pronounce it, but the acronym is EGCG, could inhibit alpha-synuclein aggregation. And unfortunately, this phase 3 trial also proved to be ineffective. There's also an agent, ATH434, that inhibits alpha-synuclein aggregation as well as reducing oxidative stress. And this is another trial that's active. It's completed recruiting and is actively gathering data. And finally, sirolimus is intended to increase alpha-synuclein autophagy, that is, that the cell where the misfolded protein is being made and aggregated would help have increased tendency to take the abnormal protein and break it down rather than letting it release. Unfortunately, this is another agent that hasn't worked out. But still, we have a number of trials that are going on and some positive signs from those trials. So trying to tackle the alpha-synuclein remains a very active target for research in multiple system atrophy. And as mentioned before, while it may not get the genie back in the bottle, we can hope that we will eventually have an agent that really slows down the progression of the fundamental aspect of the disease. If we go back to this picture, the top right, we have mitochondrial dysfunction and neurotrophic support. These are agents that are being studied. The green cell there is supposed to be the neuron. And of course, the damage to the neuron is what ultimately produces the dysfunction and problems that we see as symptoms in multiple system atrophy. So these are agents that we hope can further benefit the neurons so that they will survive any continuing production of the abnormal alpha-synuclein and its aggregates. And these agents are shown here. At the top, we start with riluzole, which is a drug that has shown some benefit in nerve survival in ALS, Lou Gehrig's disease, but unfortunately in a phase 3 randomized controlled trial in multiple system atrophy, it proved ineffective. The next two, rasagiline and safinamide, are monoamine oxidase type B inhibitors. And there's a fair bit of debatable data about to what degree these MAOB type inhibitors can have some benefit in neuron survival in Parkinson's disease. And so they were, of course, tried in multiple system atrophy as well. The rasagiline has had a phase 2 randomized controlled trial and seemed to be ineffective at slowing down the progression of multiple system atrophy. The safinamide trial is completed, seemed to be well tolerated from some initial results, but we don't have the final results from that study. Another agent that can help neurons survive is growth hormone, and that's been studied in a phase 2 randomized controlled trial, but unfortunately it also was ineffective. Now, 3 of the things that come next are mesenchymal stem cell. These are stem cells, and they can have neurotrophic effects. In this first one, stem cells were given intra-arterially and intravenously, so simple infusions into the bloodstream. And in a phase 2 randomized controlled trial, they did see a smaller increase in the total unified MSA rating score, the UMSARS, but there were safety concerns because the introduction of these cells into the bloodstream seemed to possibly cause some problems with blockages in blood flow because there were small mini stroke-like lesions seen on MRIs in the brains of about a third of the patients. So if we skip over fluoxetine a minute, the last 2 trials on this slide are also mesenchymal stem cells, but intrathecal delivery. So this would involve, for instance, putting them in through a lumbar puncture. And a small open label trial showed that this is safe and well tolerated, caused some issues in terms of side effects, but it also had a slower rate of the unified MSA rating scale progression. They didn't have a control group, but they compared it to the control group from a different drug study. So patients who were getting placebo in a drug study for multiple system atrophy, and they found that they had a slower rate of progression. So they've gone on to a larger study, phase 2 randomized control trial, and that is still recruiting at this point. So I'll go back to fluoxetine that I skipped over. And this is a fairly well-known drug used for depression, among other things, but it does show increased neurotropic support. It helps nerve cells survive. And unfortunately, it in a phase 2 randomized controlled trial was ineffective. There was not a sufficient change in the total unified MSA rating scale at 3 months. But wait, there's more. So ubiquinol, otherwise known as coenzyme Q10, has been known to be beneficial to nerve cells, and actually in a trial showed that it reduced the motor score in the UMSARS progression, so that there was some benefit demonstrated from this phase 2 randomized controlled trial. Exenatide, which is a glucagon-like peptide 1 receptor agonist. These are the Ozempic type drugs that have gotten a great deal of enthusiasm for all sorts of uses lately. Has been tried because it may help neurons to survive in patients with multiple system atrophy. And this trial actually is probably completed now, but it's still listed in the clinical trials database as active with the recruitment completed. So we don't know the results of that trial yet. This inosine 5' monophosphate can increase urate levels, which is known to reduce oxidative stress and may be beneficial to neurons. And in a phase 2 randomized controlled trial, it was demonstrated to be safe and tolerable. And there was a significant rise of uric acid, so that's proof of principle. But we've yet to see a study that actually measures whether there's significant benefit to patients with multiple system atrophy. And then we have 2 other drugs. KM819 inhibits part of the pathway that leads to apoptosis, cell death in neurons. And this is recruiting to see if it can help preserve neurons in multiple system atrophy patients. And ONO2808 is another modulator of neurotrophic effects, so it can help neurons survive. And that one's still recruiting for a phase 2 randomized controlled trial. So altogether, there's quite a few of these studies looking at neurotrophic support for the nerve cell. And that leaves us with a single gray box on the upper left that says neuroinflammation. And this is an area that I think is going to become much more exciting. This figure is taken from a review last year by the late Gregor Wenning and one of his co-workers, and starts in the upper right corner with alpha-synuclein aggregates forming in the oligodendrocytes. And so it goes down with the arrow to glial cell inclusions in oligodendrocytes and neurocell inclusions in the neuron. And the neuron, of course, stretches over towards the left side of the screen with its connections to other neurons, but it's breaking down at its distal ends. But around it, you see these orangey cells, which are activated microglia, and you see activated astrocytes. So these are other brain cells, and they become activated, and they're releasing pro-inflammatory cytokines, and T cells, which are part of the immune system, come in. And the idea is that this inflammation is a major way that the aggregates, and again, you see the little red squiggles that are supposed to be the fibrils floating around, can damage the neurons rather than strictly just by damaging the neurons directly. They induce this neuroinflammation, which damages the neurons and its connection to other cells. Now, one point to be made here, if you look at the activated microglia at the bottom of the screen, it says they're associated with alpha-synuclein clearance. And that's one of the things, these cells can actually eat up some of those little fibrils that are getting out there and may be beneficial. Whereas the activated microglia at the top of the picture have a pro-inflammatory response and are involved in generating the inflammation that may be damaging the neuron. And I'll come back to that in a moment. So these are the only agents that have been the focus of trials in multiple system atrophy, actually targeting inflammation. One is verdiperstat, which is a myeloperoxidase inhibitor and garnered a great deal of excitement in an early phase 2 randomized controlled trial. It was safe and well tolerated. There was no change in the PET binding that was supposed to show the effect on the neuroinflammation. And indeed, a much larger phase 3 randomized controlled trial actually showed that this was ineffective. There was not a significant change in the unified MSA rating scale score at 48 weeks, which was disappointing. But there may be other agents to be looked at. And then minocycline, which comes next, which inhibits that microglial activation that we were just talking about, the activated microglia. And this was ineffective in a phase 3 randomized controlled trial, but did show reduced microglial activation on PET scan imaging in an ancillary study. And that's where we come back to actually activating the microglia does two things. Some of those activated microglia are helping to chew up the abnormally folded and aggregated alpha-synuclein, but others are involved in inducing the inflammatory response. So it does beg the question, well, if you could greatly reduce the amount of abnormal alpha-synuclein folding that's going on, then any loss of benefit from not activating microglia to chew up the abnormal alpha-synuclein would be decreased, but still the blockage of the inflammatory effect from the alpha-synuclein aggregates could benefit the neurons. So you might actually shift the balance by combining a drug that affects the microglial activation with a drug that affects the production of abnormally folded alpha-synuclein. And that really speaks to one of the main points I wanted to make, which is the idea that we're probably going to need two different agents acting, one on suppressing the abnormal alpha-synuclein and two on suppressing a downstream effect that helps to damage the neuron. And it looks like the downstream effect that we really need to concentrate on is the neuroinflammation. And finally, we have intravenous immunoglobulins, again, giving infusions of antibodies, but here they're aimed at reducing the inflammatory process. And in a very small open label study, it was shown that this did result in some increases in blood pressure and some improvement in both the Part 1 and Part 2 unified MSA rating scale scores. It didn't show any changes on MRI. There were dropouts due to skin rash, but still this is a very small open label study, but with indication that there is some promise to using this approach. Unfortunately, these are the only drugs, 3 of them, that have really looked at what we can accomplish by focusing on the secondary neuroinflammation in MSA. So that brings me to my conclusions. Clinical trials of agents directed at alpha-synuclein misfolding and aggregation, which is thought to be the initial trigger for the alpha-synucleinopathies, including multiple system atrophy, are alive and well. We have 5 trials that are active, some recruiting patients, others collecting data. We have a proof of concept trial for active immunization against toxic alpha-synuclein forms that's had some positive results. And there are 3 agents that have been proven ineffective, which you always have to expect that some of the things you try aren't going to work. Similarly, in clinical trials for neuroprotective agents, this is an area that's also alive and well. 10 agents are being or have been studied in 13 different trials. 4 of the trials had positive results in the early development of these agents. 5 trials are active, either recruiting patients or processing data. 4 agents haven't worked out. But overall, the scoreboard for these 2 approaches is fairly promising. But agents directed at alpha-synuclein misfolding and aggregation, as I said, are probably more likely to slow the progression of diseases such as multiple system atrophy, but not totally halt or eliminate the pathology. Recent evidence is beginning to suggest that alpha-synuclein pathology in multiple system atrophy induces the neuroinflammation, which is responsible for a substantial part of the damage to the neurons and other brain cells. And this suggests that agents which reduce this neuroinflammation could work with the agents that inhibit the alpha-synucleinopathy to further limit the progression of multiple system atrophy. So far, only 3 agents against neuroinflammation have been studied in multiple system atrophy, and 2 of them have had negative results, though the minocycline showed some promise in an ancillary study. Intravenous immunoglobulins showed promise in a small exploratory study, but we really need more work in this area, and I hope to see this become a much more exciting area in the research on clinical trials for multiple system atrophy. Thank you for your attention.